Hello everyone. So today we're going to be talking about the de novo synthesis of both purines and pyrimidines. I know a lot of students find this pathway challenging, partly because of the number of steps, the complex enzyme names, and the interplay between different metabolic pathways. It also integrates the carbon metabolism, the nitrogen metabolism, mitochondrial function, and folate biochemistry. But today we'll break it down logically. We're going to be looking at how purines and pyrimidines are built from simple precursors, where the pentose phosphate sugar comes from, and how we get deoxyribose that we see in DNA, that deoxyribose sugar, from that ribose sugar that we see in RNA. Now, previously we explored the concept of nucleic acid polymerization, the 5 to 3 prime pattern, and DNA replication, and we looked at how nucleotides are incorporated into a growing strand of DNA during DNA replication, and we also looked at how nucleosides undergo stepwise phosphorylation to become the activated triphosphates required for DNA synthesis, that daughter strand synthesis, as well as RNA synthesis. Now, today we're going to be taking a step back from that endpoint, and we're going to be examining where these molecules actually come from. We'll look at how ribose sugars are generated through the central carbon metabolism and the hexomonophosphate shunt. We'll look at how nitrogen atoms are donated from various amino acids like glutamine and aspartate to kind of contribute towards building these purine and pyrimidine nitrogenous rings. And by the end, you should not only understand the steps, but also kind of see the bigger picture of where this kind of fits into, DNA replication, stepwise phosphorylation, and we'll also understand the pentose phosphate shunt and the difference between purine synthesis and the difference between de novo pyrimidine synthesis. So the de novo synthesis of DNA is a crucial process to cells functioning properly. So as you know, cells need to divide, cells need to repair their DNA, and that those mechanisms require sufficient amounts of deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxyuridine triphosphate, and thymidine triphosphate. Okay, and these small imbalances can have detrimental effects on the cell itself. As you know, for example, DNA synthesis, when synthesizing our daughter strand of DNA and replication, it's really important to have a sufficient amount of free nucleotide triphosphates, pools of nucleotide triphosphates to kind of draw from. And if we don't, it can have serious downstream effects such as mutagenesis. It can cause genetic mutations in DNA. It can cause replication fork stalling if that daughter strand isn't being formed quick enough, and it can also contribute towards chromosomal instability. So for those reasons, because of those implications, it has to be a very tightly regulated process, and it's coordinated with the cell cycle. So especially the S phase, or the synthesis phase of the cell cycle, when our cells are getting ready to divide before mitosis, they're going to be doubling up, or they're going to be replicating their entire genome. So you can expect, because of those reasons, that these metabolic processes that are associated with the de novo synthesis of both purines and pyrimidines is going to be upregulated. And it's also very metabolically expensive. It requires ATP as a phosphate donor for many of the metabolic intermediates, and it also requires NADP availability as well. So it kind of interplays with our energy metabolism as well, especially when you look at the formation of the ribose or deoxyribose sugar you see in both RNA and DNA. There's an interplay there between the hexomonophosphate shunt, or the pentophosphate shunt, and glycolysis itself. So depending on cellular needs, and ATP might determine where those where that ribulose or ribose 5-phosphate will be directed towards. It will either feed back into glycolysis, or it will fully commit to the de novo synthesis of purines and pyrimidines by becoming phosphoribosyl pyrophosphate. So just to recap on what our cells are actually building here. So by de novo, we mean from scratch, and that's a term that's used in biochemistry a lot, de novo synthesis. Now, just to recap, DNA itself is a polymer or a polynucleotide, so it's made up of many repeating chemical subunits, and those repeating chemical or monomeric subunits are referred to as mononucleotides. Now, we know mononucleotides, if we're looking at them as just a unit themselves, they contain a nitrogenous base, which is that heterocyclic aromatic ring, and that's either a purine or a pyrimidine. that nitrogenous base is bonded to a deoxyribose sugar in the case of DNA via a glycosidic bond, and that fifth carbon on the deoxyribose sugar is bonded to a phosphate group. Now, I already went over this in a previous video, so if you're a bit confused, there is another video looking at this in more detail. Now, as I said before, those nitrogenous bases that partially make up that mononucleotide unit, if you like, can either be divided into two chemical families. One of them are the purines, and that consists of adenine and guanine. And we know purines have that imidazole ring and they have that six-membered pyrimidine ring. And both those imidazole and pyrimidine rings are heterocyclic. Okay, and then we have our pyrimidines, which consist of thymine, cytosine, and if we're looking at RNA, uracil. Now, our pyrimidines are much more simplistic in structure, and they just have a six-membered pyrimidine ring, which is also heterocyclic. Now, there's a critical point to kind of remember here before we move on, is that both, depending on whether you're synthesizing a purine mononucleotide or a pyrimidine mononucleotide, will completely will dictate two entirely different synthetic mechanisms. The first one we're going to talk about is the synthesis of purines, and then we're going to talk about the synthesis of pyrimidines. So purines themselves are built on what's called a ribose scaffold. So I mentioned previously that ribose 5-phosphate will either fully commit to de novo nucleotide synthesis by becoming phosphoribosyl pyrophosphate, or ribose 5-phosphate will feed back into glycolysis by becoming an intermediate like glyceraldehyde 3-phosphate. Now, purines, because of their complex structure and that imidazole ring, they require phosphoribosyl pyrophosphate to be used as a sugar scaffold, if you like, a ribose scaffold. So they're built directly onto the sugar. But pyrimidines are built slightly differently. Pyrimidines, the ring itself is built first, and then it's attached onto our phosphoribosyl pyrophosphate. So just to recap, purines are built using a ribose scaffold, and pyrimidines are built as free rings and attached onto that ribose sugar. And we're going to look at purine synthesis first. We're going to look at how that happens. So I'm going to talk about the pentose phosphate shunt first, because to get our phosphoribosyl pyrophosphate, first glucose 6-phosphate has to be shunted from glycolysis and directed towards the pentose phosphate pathway to become our phosphoribosyl pyrophosphate, to use that as a sugar scaffold to build our purine base from or onto. So as I said before, purines have a much more complex ring structure or complex structure than their pyrimidine counterparts. Purines are these complex multi-atomic rings. They've got that imidazole ring and they've got that pyrimidine ring. So if you assembled them free floating like you do pyrimidines, it would be a lot more complicated. They're more unstable and it could contribute more to toxicity. So it's important that we use that our ribose sugar, our PRPP, or our phosphoribosyl pyrophosphate, as a scaffold to kind of provide some kind of structural anchoring and spatial orientation. Okay, and it also stabilizes that purine ring a lot more. So as I said before, glucose 6-phosphate is not necessarily metabolically committed to glycolysis, but a lot of people call it the pentose phosphate shunt because they see it, because when energy or when ATP demands in the cell aren't as high, a lot of glucose 6-phosphate is shunted towards the pentose phosphate pathway. So that glucose 6-phosphate will undergo, or its metabolic fate will be completely different to that of what it will be in glycolysis. Now, the pentose phosphate pathway has the ability to feed back into glycolysis if necessary, which I'll talk about later, which is also a really important concept to understand when looking at ATP, kind of balancing ATP, balancing cellular needs, and looking at the metabolism more holistically rather than just these kind of separate isolated pathways. Now, glucose 6-phosphate will eventually be used to form ribose 5-phosphate, and then ribose 5-phosphate can be converted into phosphoribosyl pyrophosphate, and that phosphoribosyl pyrophosphate is really important in acting as our ribose scaffold to build our purine nitrogenous base from. So intracellular glucose is activated or metabolically activated by phosphorylation to form glucose 6-phosphate, and the phosphorylation of intracellular glucose commits it to metabolism. Doesn't necessarily commit it to glycolysis or the pentose phosphate shunt, but it commits it to metabolism. Now, the phosphorylation of glucose is catalyzed by the enzyme glucokinase in hepatocytes and beta pancreatic cells, or hexokinase in other body cells. Now, both those enzymes will use ATP as a phosphate donor, giving you ADP, okay, and that phosphate group is transferred onto the 6th carbon of the hexose ring, hence the name glucose 6-phosphate. And that phosphate group is very negatively charged, so it's going to keep glucose in the cell. It's going to prevent it from moving through those GLUT transporters or GLUT protein transporters that are embedded in that lipid bilayer of our cell membranes, and that kind of controls diffusion chaos, if you like, and keeps the glucose 6-phosphate in the cell. The phosphorylation also makes it more susceptible to, say, redox chemistry and enzymatic activity, allowing for it to be metabolically processed, if you like. So glucose is phosphorylated into glucose 6-phosphate, and that's by either glucokinase or hexokinase using ATP as a phosphate donor. And by adding that phosphate group onto the 6th carbon of the hexose ring, you're essentially trapping glucose in the cell. Those GLUT4 transporters and GLUT2 transporters rely on passive diffusion chemistry, so it's not a really controlled process. So what can happen is for every glucose molecule going into the cell, naturally you can get a glucose molecule diffusing out of the cell. But by adding those negatively charged phosphate groups, you're preventing that passive diffusion, because these GLUT4 transporters don't take large charged molecules, and you've added that big negative phosphate group onto it. Now, of course, it also makes the molecule itself more susceptible to enzymatically controlled reactions, or enzymatic isomerization, redox chemistry, which essentially drives a lot of these metabolic pathways. You're altering that electron distribution on that hexose ring. Now, of course, glucose 6-phosphate itself is essentially a metabolic hub, so it's capable of feeding into many different pathways depending on where you are in the body and depending on cellular needs. So, for example, glucose 6-phosphate can be converted into glucose 1-phosphate by phosphoglucomutase, and that glucose 1-phosphate is really important in glycogenesis in your liver tissue, in your muscle tissue, and in your fat tissue. Of course, when blood sugar levels get too high, you're going to need to store some of that glycogen or some of that glucose in the form of glycogen in the liver, okay, because glycogen is a polymer of glucose. And of course, in fat cells as well, fat cells need those glycogen reserves to be able to synthesize their glycerol for their triglycerides. And of course, muscle cells need glycogen for quick access to ATP to meet energy demands, especially during high intensity training. Of course, glucose 6-phosphate can also feed into glycolysis, and that's really important as well, because glycolysis is essentially a process that allows for the production of ATP, the generation of pyruvate, okay, and the production of even more ATP and NAD. But also glycolysis is another biosynthetic hub, so a lot of those metabolic intermediates can then go on to fatty acid synthesis. They can be used to synthesize, like phosphonyl pyruvate can be used to synthesize glucogenic amino acids like alanine. So of course that's really important as well. And that adding that phosphate group allows for that redox chemistry and that isomerization chemistry to happen, because of course glucose 6-phosphate is converted into fructose 6-phosphate, okay, and fructose 6-phosphate, that isomerization reaction is only feasible with that phosphate group on glucose, because these enzymes are responding to charge and geometry, so they're responding to glucose 6-phosphate, and then of course that fructose 1,6-bisphosphate is then split into glyceraldehyde 3-phosphate and dihydroxyacetone, okay, and that's a really important step in glycolysis, because that initiates the payoff phase where you actually generate ATP and your reduced coenzymes NAD, and of course you get your 2 molecules of pyruvate as well. But of course glucose 6-phosphate can be shunted to the hexomonophosphate pathway, so the addition of that phosphate group makes it more susceptible to enzyme-specific oxidation, for example. It's really important in committing that glucose 6-phosphate is oxidized into 6-phosphogluconolactone, and it's fully committed to the oxidative phase of the pentose phosphate pathway, which we'll talk about in the coming slides. So you can kind of see the importance of glucose 6-phosphate. It's called a metabolic hub. okay, because it has several different metabolic fates. And glucose 6-phosphate is often associated with the pathway glycolysis, and glycolysis itself is considered a biosynthetic hub too. But of course, I'll talk about that in another video. Today we're focusing on DNA synthesis. But of course, that glucose 6-phosphate, when it's shunted to the hexomonophosphate shunt and is oxidized into phosphogluconolactone, it's really important to remember that that phosphogluconolactone, or that 6-phosphogluconolactone, is not fully committed to DNA synthesis yet, but or purine, de novo purine pyrimidine synthesis, it's fully committed to the oxidative phase of the hexomonophosphate shunt, so it's fully committed to becoming ribose 5-phosphate. But of course, it's not necessarily fully committed to de novo purine and pyrimidine synthesis until that ribose 5-phosphate is converted into phosphoribosyl pyrophosphate by your phosphoribosyl pyrophosphate synthetase enzyme. So the pentose phosphate pathway is also called the glucose 6-phosphate shunt or the hexomonophosphate shunt. So it has different names depending on what literature you're reading and what who's teaching you essentially. But it's all the same pathway. It just has different names. So don't be thrown off by that. A lot of people get confused. But the pentose phosphate pathway is a cytosolic metabolic pathway that oxidizes glucose 6-phosphate to generate NADPH, that coenzyme NADPH, and ribose 5-phosphate. And that ribose 5-phosphate can then be can then become a phosphoribosyl pyrophosphate, catalyzed by the enzyme PRPP synthetase, and that phosphoribosyl pyrophosphate is what we either attach our pyrimidine onto once the pyrimidine ring has been assembled, free floating, if you like, or we can use that phosphoribosyl pyrophosphate to act as a kind of sugar scaffold to build our purine nitrogenous base from. Now, it's important to know to kind of acknowledge that the pentose phosphate pathway and glycolysis essentially run parallel to each other, and there are several reasons for this. Okay, the main reason being that the pentose phosphate pathway does not produce ATP. It's anabolic and regulatory, whereas glycolysis does. So it's not an energy yielding process, but we need ATP to act as a phosphate donor, okay, to form several of our metabolic intermediates, because those phosphate groups are going to activate certain atoms like our carbon or our oxygen to make it more susceptible to chemical reactions, to act as a good leaving group, if you like. But if the energy levels aren't sufficient in the cell and we need more ATP, what our cells can do is they can take those intermediates, that ribose 5 from the pentose phosphate pathway, and feed it back into glycolysis. And I'll get into that later when we look at the oxidative versus the non-oxidative phase of the pentose phosphate pathway. And because we're looking at the de novo synthesis of nucleotides and we're not really looking at energy pathways, we're not really looking at glycolysis, essentially, we're going to mainly focus on the oxidative phase, or the first phase of the pentose phosphate pathway, which gives you ribose 5-phosphate. So glucose 6-phosphate, you get ribose 5-phosphate, you go from a 6-carbon sugar to a pentose 5-carbon sugar. Okay, and that ribose 5-phosphate can either fully commit to de novo synthesis of DNA, but it will only fully commit to that de novo synthesis when it becomes phosphoribosyl pyrophosphate. Okay, if not, it can feed back into glycolysis. It can undergo reactions. It can become glyceraldehyde 3-phosphate, and as you know, glyceraldehyde 3-phosphate is another metabolite further downstream of glycolysis. Once that fructose 1,6-bisphosphate is split into either dihydroxyacetone and glyceraldehyde 3-phosphate, and I'll show you how that kind of happens as we move on into the slides. But for now, all you need to know, the main takeaway, is that the pentose phosphate pathway runs parallel to glycolysis. That glucose 6-phosphate that initiates glycolysis can be shunted into the pentose phosphate pathway, and then our metabolites, ribulose 5-phosphate and ribose 5-phosphate, can feed back into glycolysis. And then essentially glycolysis, as you know, will generate ATP. So depending on the cellular needs, depending on ATP needs and energy needs, will depend on the fate of that ribulose or ribose 5-phosphate. So the pentose phosphate pathway is initiated by the conversion of glucose 6-phosphate into 6-phosphogluconolactone, and this is an oxidation reaction. So glucose 6-phosphate is shunted from glycolysis, okay, and then the enzyme glucose 6-phosphate dehydrogenase, along with the cofactor nicotinamide adenine dinucleotide phosphate, or NADP+, is going to oxidize our glucose 6-phosphate, the hydroxyl group on that first carbon of the hexose ring on our glucose 6-phosphate. And this oxidation results in the formation of a carbonyl group on that first carbon, giving us 6-phosphogluconolactone. Now our NADP plus is taking those 2 electrons and 1 hydrogen. Okay, so it's becoming reduced into NADPH, and that NADPH is a really important coenzyme downstream in these metabolic pathways, particularly the formation of our deoxyribose sugar. That hydroxyl group, or that additional oxygen our ribose sugar possesses, has to be removed to get our deoxyribose sugar, and our NADPH is going to do that oxygen removal by radical chemistry or redox chemistry. So it's acting as a oxidizing or agent, and it itself will form NADP plus again in that process. So the generation of our NADPH is really important to kind of take away from this. Okay, so glucose 6-phosphate has been oxidized into 6-phosphogluconolactone, and our NADP plus has been reduced. Okay, so it's taken those 2 electrons and that hydrogen proton. Okay, and you get a plus H plus, you get a hydrogen proton left in solution, because the NADP plus can only take 2 electrons and 1 hydrogen proton. It can't take 2, so it leaves 1. Now that formation of that carbonyl group, okay, will change that changing electron distribution will cause a ring change as well. So you get what's called a lactone ring, and that lactone ring is a cyclic ester, okay, and it's formed by the oxidation of a hydroxyl group into a carbonyl group, followed by intramolecular esterification. So step 1 was an oxidation reaction. We had our glucose 6-phosphate, and that hydroxyl group on the first carbon was oxidized into a carbonyl C double bond O group, and that gave us our phosphogluconolactone, and we had that lactone ring formed. But the next step is conversion of phosphogluconolactone into 6-phosphogluconate. Now, this is a hydrolysis reaction, and this is a really important reaction. Now, this hydrolysis reaction essentially adds water, and the enzyme that catalyzes this reaction is 6-phosphoglucolactonase, and this still occurs in the cytosol. Now, this hydrolysis results in the formation of a carboxyl group, or a carboxyl functional group at the first carbon, and that's a COO minus group. Now, this change, this rearrangement of electron distribution essentially causes that lactone ring to form a linear molecule. So that ring is broken. Okay, entropically we now favor that linear state rather than our cyclic state. Now the formation of this carboxyl group primes it for oxidative decarboxylation. Now, oxidative decarboxylation is really important in removing that carbon, because the whole concept of the pentose phosphate shunt is we're going from a 6-carbon or a hexose sugar to a 5-carbon pentose sugar, and the way to remove that carbon is through oxidative decarboxylation. But in order to prime our intermediate for that process, we need to add that carboxylic acid group, and in order to do that we need a hydrolysis reaction. We need to add our water, and now we've also alleviated ring strain as well, that lactone. The molecule's more ready for enzymatic oxidation or oxidative decarboxylation, being in that linear state. Now, something that I didn't mention before, and this goes the same with the previous step. So when we had our glucose 6-phosphate, we oxidized that hydroxyl group into that carbonyl group, and we got our phosphogluconolactone. And now we've undergone a hydrolysis reaction, and now at that first carbon we've gone from a carbonyl group to a carboxyl group, or a carboxylic acid group. And these steps, they're all irreversible reactions, but we're still not fully committed to de novo synthesis of our nucleotides. Now, we don't fully commit to nucleotide metabolism until we get our activated ribose sugar, our phosphoribosyl pyrophosphate. But what we have ensured is that these metabolites are committed to the oxidative phase of the pentose phosphate shunt, and the end of the oxidative phase marks the formation of our ribulose or our ribose 5-phosphate. And depending on cellular requirements, that ribose 5-phosphate can feed back into glycolysis. So we're committed to the oxidative phase of the pentose phosphate pathway, but we're still not fully committed to de novo synthesis. And the main important takeaway from this hydrolysis step is we've primed, in forming our 6-phosphogluconate, we've primed the molecule or the intermediate for oxidative decarboxylation and the formation of a ribose sugar. We're removing that carbon from our hexose or our six carbon compound to a 5-carbon compound. So our cells have now primed our 6-phosphogluconate metabolite for oxidative decarboxylation. And how did we achieve that? We achieved it through a hydrolysis reaction of phosphogluconate lactone. So that carbonyl group that we formed from the previous oxidative step became hydrolyzed. We added water and we got our carboxyl functional group. Okay, so we're now primed for that oxidative decarboxylation. The removal of that functional group, okay, will give us a template or a foundation for our 5-carbon sugar. So 6-phosphogluconate undergoes oxidative decarboxylation, and the enzyme that catalyzes this reaction is 6-phosphogluconate dehydrogenase, or it's also called oxidoreductase. Okay, and it uses that cofactor NADP+. Now, before we were focusing on that C1 carbon on our molecule, but now we're going to go to our third carbon. Now, our third carbon contains that hydroxyl group, okay, and our cofactor NADP+ is going to oxidize that hydroxyl group into a ketone, okay, and a ketone is still a C double bond O functional group like that carbonyl group. The only difference is that carbonyl group, a carbonyl functional group, exists on the N-terminal of a molecule, okay, whereas ketones, if you like, are carbonyl groups that are embedded within the carbon chain. They're not on the N-terminus, if you like. So the oxidation of that hydroxyl group on the third carbon gives us that ketone group. We get a beta keto intermediate, and that's unstable and prone to decarboxylation. So that carboxyl group on the first carbon, entropically it's going to leave. It wants to leave because the molecule's unstable. You get spontaneous decarboxylation, okay, and this results in our ribulose 5-phosphate, and that serves as a template that carbon group has left. Okay, that carbon's left, giving us a 5-carbon compound, which will serve as our kind of foundation for our ribose sugar. Now, when you get the formation of ribulose 5-phosphate, you also get spontaneous cyclization, because entropically it favors being in that cyclic ring, and that's because there's an intramolecular nucleophilic attack. There's a hydroxyl group on that phosphoric acid group at the N-terminus that's going to attack that newly formed carbonyl group or ketone group, if you like, and it's going to essentially fold in on itself and form this cyclic ring. And that is essentially the formation of our 5-carbon ring. So we've now gone from a 6-carbon compound, and to be precise, we've gone from a hexo sugar or a 6-carbon sugar to a 5-carbon ribose sugar. So now we have our ribulose 5-phosphate, we need to convert it into ribose 5-phosphate, and ribose 5-phosphate is an isomer of ribulose 5-phosphate. So they have the same chemical formula, the same amount of carbon, hydrogen, oxygen and phosphorus make up their chemical structure, if you like. They're just arranged in a slightly different space, which is essentially what an isomer is, the same way, you know, fructose is an isomer of glucose. They have the same chemical formula, just a different spatial arrangement. Now, how did we get our ribulose 5-phosphate? Okay, well, that was through an oxidative decarboxylation of our phosphogluconate. Okay, and that oxidation of that third carbon, that hydroxyl group on the third carbon, by our NADP plus as our oxidizing agent, resulting in the formation of a keto group. It forms an unstable beta keto intermediate, okay, and then that carboxyl group on that C1 leaves, okay, getting CO2 as a byproduct. Now we have our ribulose 5-phosphate, and the enzyme phosphopentose isomerase or ribose 5-phosphate isomerase will convert ribulose 5-phosphate into ribose 5-phosphate. Now, both DNA and RNA synthesis require ribose 5-phosphate because ribose 5-phosphate can be readily converted into phosphoribosyl pyrophosphate, which, as you know, acts as our ribose scaffold in purine synthesis, and it's directly attached to our readily assembled pyrimidine ring in pyrimidine synthesis. Now, essentially, what's happening? What the difference is? So, ribulose 5-phosphate, for example, has that ketone group in its third carbon, whereas ribose 5-phosphate has an aldehyde group, an aldehyde functional group, a C double bond H group. Okay, and it's N-terminus. You can see that I've compared them, and I've given a linear example, but it's important to remember that canonically in the cell they will be in their cyclic or their ring structure, their pentose ring structure. Now, the enzyme initially forms an enediol intermediate and it rearranges to form an aldehyde at that C1 to get you ribose 5-phosphate. And then ribose 5-phosphate is a precursor for our PRPP or phosphoribosyl pyrophosphate, which is the activated sugar for our nucleotide synthesis. Now, it's important to know that ribose 5-phosphate is still not metabolically committed to de novo DNA synthesis because depending on cellular needs, ribose or ribulose and ribose 5-phosphate can enter transketolase or transaldolase reactions and feed back into glycolysis. So they can be used to form intermediates, like, as I said before, glyceraldehyde 3-phosphate, which can directly feed back into glycolysis. It's not until we get the formation of our phosphoribosyl pyrophosphate that we're fully committed to de novo nucleotide synthesis. That is our activated sugar. Now, I'm showing you here a path up the whole pentose phosphate pathway. Now, for de novo DNA synthesis and for purine synthesis, we looked at the oxidative phase. So if we went from glucose 6-phosphate, okay, we did an oxidative reaction using NADP plus as a coenzyme to get us 6-phosphogluconolactone. And how did we do that? What actually happened mechanistically speaking? That hydroxyl group on the first carbon was oxidized into a carbonyl group. Okay, and the second phase was a hydrolysis reaction catalyzed by our enzyme gluconolactonase. Okay, and that hydrolysis reaction at the C1 allowed for the formation of a carboxyl or a carboxylic functional group, a carboxylic acid functional group to be precise, and that's a COO minus group. And it can also be canonically COOH, but COO minus is also a carboxyl functional group. Okay, and we've primed it for oxidative decarboxylation. So in the next phase, when we get our 6-phosphogluconate, which has that it has that carboxyl group, if you like, it can form ribulose 5-phosphate through a oxidative decarboxylation reaction, okay, using NADP plus as a coenzyme. So our 6-phosphogluconate dehydrogenase along with NADP plus has taken those two electrons and those two hydrogens away from that molecule, particularly that third carbon. That third carbon initially had a hydroxyl group and it now has a keto group, a C double bond O group embedded within the molecule. Okay, and that creates an unstable beta keto intermediate, and that carboxyl group is going to leave through decarboxylation. It's more stable, removing that CO2 makes the molecule more stable. Okay, now we've got our 5-carbon compound ribulose 5-phosphate. Ribulose 5-phosphate is going to undergo cyclization because that ketone group is now going to act as a nucleophile, if you like, and we've got our hydroxyl group that's on the phosphoric functional group at the other end of the molecule. It's going to fold in on itself in an intramolecular reaction, forming our ribose 5 or our pentose ring. Then we have bleeding into the kind of non-oxidative phase, we have the formation of our ribose 5-phosphate, and that was an isomerase reaction. Okay, we converted ribulose 5-phosphate into its isomer ribose 5-phosphate, and then what's going to happen then is PRPP synthetase is going to donate 2 phosphates onto our ribose 5-phosphate, forming phosphoribosyl pyrophosphate, and that's our activated sugar that's fully committed to the de novo synthesis of our nucleotide bases. But what you're seeing on the right hand side is the pathway, or the pentose phosphate pathway, that forms the intermediates that will feed into glycolysis. So as I said before, those transketolase reactions, okay, or that will form your glyceraldehyde free phosphate, and that glyceraldehyde free phosphate can feed into glycolysis, or you can form your xylulose 5-phosphate and your acetoheptulose 7-phosphate. Okay, and a transaldolase reaction can give you your fructose 6-phosphate, which can feed back into glycolysis as well. So you can see how you can kind of see how these pathways will feed into glycolysis or feed into de novo synthesis of DNA, depending on what the cell needs or what the cell requires. So now we're at what's called the commitment step, and as I said before, ribose 5-phosphate is not necessarily committed to de novo purine or pyrimidine synthesis. It's only committed when it becomes phosphoribosyl pyrophosphate. Okay, and this is a phosphoryl transfer activation reaction. It's catalyzed by the enzyme PRPP synthetase. Okay, and magnesium is used as a cofactor again, and ATP is used as a phosphate donor. We're transferring that beta and gamma phosphate directly onto our ribose 5-phosphate, and we're using magnesium to stabilize that enzyme substrate complex to prevent that natural electrostatic repulsion that's going to be felt from those negative phosphate groups. Okay, and the addition of pyrophosphate to that C1 increases its ability as an electrophile. Okay, and that the addition of that pyrophosphate creates a good leaving group. Okay, it wants to leave, it makes it more chemically reactive, if you like. So now we've got our phosphoribosyl pyrophosphate, we're fully committed to the de novo synthesis. Okay, we've finished we're bleeding into the non-oxidative phase of pentose phosphate pathway. We're not going to see it through. Okay, so all the other metabolic intermediates you saw before that, like glyceraldehyde 3-phosphate and fructose 6-phosphate, we're not going to be used to form that. We're going to be used to form DNA, and I promise I will go over the rest of the pentose phosphate pathway when we're looking at glycolysis, red blood cell metabolism, and all of that. Now, pyrophosphate is a large kind of charged and stabilized molecule, and upon release it wants to be on its own. Okay, upon release it's very stable, so it allows that C1 carbon to be primed for a nucleophilic attack, or to act as an electrophile. Okay, by phosphorylating it, by adding those two phosphates, our pyrophosphate, we've created a good leaving group that actually wants to get out. You know, it's not happy where it is, and we've made our compound, we've made ribose 5 more chemically reactive by converting it into phosphoribosyl pyrophosphate, ready to assemble our purine base from, using it as a scaffold, or using it to directly assemble our already formed pyrimidine base onto. So now we've got our phosphoribosyl pyrophosphate, we can start using that as a ribose scaffold to build our purine ring from. Now, as you know, purine rings are heterocyclic in nature, so they're made up of both nitrogen and carbon. So we're going to need nitrogen donors. Okay, and if you remember as well the structure of a purine, that imidazole ring, that 5-membered imidazole ring, the nitrogen on that 5-membered ring directly bonds to the deoxyribose sugar through a nitrogen bridge or a glycosylic bond. Okay, so we're forming that first nitrogen that's bonded to the deoxyribose sugar. Now, the enzyme that catalyzes this reaction is referred to as glutamine phosphoribosyl pyrophosphate amidotransferase, and it's also called amidophosphoribosyl transferase. So it's important to look out for the different names of enzymes in literature. They're still the same enzyme. Now, structurally, it has two domains. It has the glutamine amidase domain, and that performs the amine hydrolysis on our glutamine. Okay, because we need to isolate that amine group. We want to attach that amine group from glutamine onto our phosphoribosyl pyrophosphate. And it also has a PRPP, or a phosphoribosyl pyrophosphate transferase domain, and that's where the amine group is fixated onto our ribose sugar. Now, it's important to acknowledge that those two functional domains are connected to by what's called an internal ammonia channel, or an internal hydrophobic ammonia channel. As you know, amine groups will form ammonia once dissociated, and ammonia is incredibly toxic to cells. Nitrogen is toxic to cells, and so is free floating amine groups. So this internally ammonia channel prevents ammonia exposure to cells. It prevents ammonia toxicity, and that's an important concept to kind of grasp when looking at a lot of these enzymes involved in this nitrogen fixation. If you like, it is acknowledging that structure and the purpose of that structure, especially when looking at purine and pyrimidine synthesis, especially purine synthesis. Anything where there's going to be a nitrogen donor and use of an amino acid, there's likely going to be an internal ammonia channel to channel that ammonia group through the enzyme into the other domain where it can be fixated onto the ribose sugar. So the glutamine amidase domain will perform a hydrolysis reaction on glutamine, okay, and that will give you glutamate and your ammonia. And of course, that ammonia is not free floating. As I said before, it's traveling through that internal ammonia channel to the phosphoribosyl pyrophosphate transferase domain, where that transferase domain is going to fixate it or put it onto that first carbon that has that pyrophosphate, okay, and that first carbon's been activated by that pyrophosphate because the pyrophosphate's a leaving group, okay, it's going to leave, and then nitrogen, that amine group, is going to be put onto that first carbon, and this gives you 5-phosphoribosyl amine, and that's our first intermediate, if you like, our first step that contains the nitrogen that's going to eventually be used as a building block to form the rest of our imidazole and pyrimidine ring on when looking at our kind of purine assembly. So I've got the equation here as well, because sometimes in exams you may be asked to break the equation down a bit more. As you know, there's a lot happening here. So we've got our phosphoribosyl pyrophosphate plus our glutamine, our amino acid glutamine, which is acting as our nitrogen donor, and the enzyme that's catalyzing this reaction again is glutamine phosphoribosyl pyrophosphate amidotransferase, okay, or amidophosphoribosyl transferase, and that's going to transfer that. It's going to hydrolyze that amine group from that glutamine forming glutamate, and then it's going to transfer that amine group through the PRPP transferase domain onto our phosphoribosyl pyrophosphate, onto that first carbon. That pyrophosphate is going to leave because it's a leaving group, okay, and you're going to get your 5-phosphoribosyl amine. So in the equation you can see I've got PRPP plus glutamine gives you 5-phosphoribosyl amine plus glutamate and pyrophosphate. So the byproducts essentially are glutamate and pyrophosphate. So as you know, the purine ring that we aim to assemble, or our cells aim to assemble, is heterocyclic, so it contains both nitrogen and carbon. So this step will incorporate both nitrogen and carbon into that initial building block. Now 5-phosphoribosyl amine is converted into glycinamide ribonucleotide. Now this is catalyzed by the enzyme phosphoribosyl glycinamide, and it's an ATP dependent enzyme because you need the ATP, the phosphate from the ATP, to activate your glycine. Okay, so the amino acid glycine is acting as a nitrogen and carbon donor, or a nitrogen and carbon building block to the ribose scaffold. So the ATP will activate the glycine. It forms a high energy glycyl phosphate intermediate, and that makes it more chemically reactive. Okay, that phosphate is added to the carboxylic acid group you see on the glycine. You can see it's got a COOH group. Okay, so a carbonyl group and a hydroxyl group makes a carboxylic acid group. Okay, and that phosphorylation makes it a better electrophile. Okay, it makes it more susceptible for nucleophilic attack. Okay, and the nucleophilic attack is coming from that amine group. Okay, because our amine is acting as our nucleophile in this case. So once we form our high energy glycyl phosphate intermediate, so that phosphate is attached to that carboxylic group, that amine group on the 5-phosphoribosamine is going to attack that phosphorylated carboxylic group, and the phosphate is going to leave as inorganic phosphate, and you're going to get your glycinamide ribonucleotide. So you've now incorporated that glycine structure into your 5-phosphoribosyl amine, okay, so to give you glycinamide ribonucleotide. So before we kind of move on to the next steps, I just want to reinforce or introduce this concept of multifunctional or trifunctional or bifunctional enzyme protein complexes. A lot of these metabolic intermediates we're dealing with here are actually highly toxic. So they are for it to reduce cell toxicity and to kind of favor metabolic efficiency, eukaryotic cells have developed these very sophisticated protein complexes, if you like, these multifunctional or trifunctional enzyme complexes that have multiple domains and allow for efficient conversion from one metabolite intermediate to another. And the first, and you'll see quite a few multifunctional and bifunctional enzyme complexes as we move down this pathway. But the first one that we're going to talk about is the GARP protein complex, and that stands for the glycinamide ribonucleotide transformylase complex, and that has multiple domains. It's a trifunctional enzyme, so it has 3 functional domains, and they're involved in the next 3 steps that I'm going to talk about in the next slide. I just wanted to kind of get everyone in with learning the idea of kind of recognizing these enzymes we're talking about as one protein complex with different domains. So the first domain we're going to talk about on the GARP protein complex is the GAR transformylase domain, and that catalyzes the conversion of glycinamide ribonucleotide to formyl glycinamide ribonucleotide. And this is a formylation reaction where adding a formyl functional group, and our formyl donor is our N10 formyl tetrahydrofolate. So that's actually our formyl donor. And essentially what's happening is that amino group on the N terminal of the glycine will attack the formyl carbon on the N10 formyl tetrahydrofolate, okay, and that CHO group is transferred to the amine group, and this forms formyl glycinamide ribonucleotide. Okay, so that formyl, that carbon on that formyl group contributes to the 8th carbon you see in your purine nitrogenous base. Okay, and that glycine's contributed to that 4th, 5th, and that 4th and 5th carbon and that 7th nitrogen. So in our previous step, we got our formyl glycinamide ribonucleotide from a formylation reaction of glycinamide ribonucleotide using N10 formyl tetrahydrofolate as our formyl or CHO donor to give us our formyl glycinamide ribonucleotide. Now, I mentioned before the GARP protein complex. Well, the next domain that we're going to talk about is the FGAM synthetase domain, or the phosphoribosyl formyl glycinamide domain. Okay, and once we get our formyl glycinamide ribonucleotide, it's shifted from the FGAR domain to the FGAM domain, okay, and that FGAM domain is going to transfer essentially an amine group from glutamate onto our formyl group on our formyl glycinamide ribonucleotide. So of course our FGAM synthetase domain has a amid hydrolysis or hydrolytic properties, is able to isolate that amine group from glutamine, giving you glutamate. Now it's an ATP dependent enzyme. The ATP, we're going to need to phosphorylate the carbonyl group on our formyl glycinamide ribonucleotide to make it more electrophilic, to allow for that NH3, that amine group that we've got from our glutamine, to bond to that carbonyl carbon at the formyl group on our formyl glycinamide ribonucleotide, and that will give us formyl glycinamidine ribonucleotide. Now, in the equation, I've got FGAR, which is formyl glycinamide ribonucleotide, plus glutamine, because glutamine is our nitrogen donor, plus ATP, because ATP is our temporary phosphate donor. We want to donate one of those phosphates to our formyl glycinamide ribonucleotide to make it more chemically reactive, okay, to allow for that amine group to act as a nucleophile, come and form our FGAM, or our formyl glycinamidine ribonucleotide. Okay, and the products are our FGAM, and FGAM just stands for formyl glycinamide ribonucleotide, plus glutamate, plus adenosine diphosphate, plus inorganic phosphate, because as we know, those phosphates are used as leaving groups. So they upon the amine group binding with that carbonyl carbon at the formyl group, that inorganic phosphate that's been transferred from ATP will leave. So we'll get that as a byproduct. So it's really important to include that when you're doing these chemical equations to kind of show you understand what's being used and what's being changed and what's being given off as a byproduct. So we had our full-meal glycinamide ribonucleotide, and we transferred an amine group using glutamine, the amino acid glutamine, as a nitrogen or amine donor, okay, to give us full-meal glycinamidine ribonucleotide. Now the next step is to form our amino-imidazole ribonucleotide, or our AIR compound, and that is the first cyclic ring, if you like, that's formed in purine synthesis, that imidazole ring. And if you know your purines, you can start to see it coming together now. That imidazole ring is directly linked to the deoxyribose sugar on a nucleotide monomer. And if you're looking at purines in particular, because they have that imidazole ring, that 6-membered pyrimidine ring is attached to that imidazole ring. Okay, so the next step is, after this, is to eventually start building that pyrimidine ring. But to take a few steps back, full-meal glycinamidine ribonucleotide undergoes a cyclization reaction, so an intramolecular cyclization reaction to form amino-imidazole ribonucleotide. Okay, so it's got that first imidazole ring. Now this is catalyzed by the enzyme amino-imidazole ribonucleotide synthetase, but it's also called full-meal glycinamide ribonucleotide cyclase. And it also has another name, phosphoribosyl amino-imidazole synthetase. So it's got 3 different names, but they're all the same domain and they do the same thing. Just someone's decided to confuse the hell out of everybody and give them 3 different names in the literature. So if you see those names, it's the same enzyme domain. So this is still part of the GART protein complex, and it's an ATP dependent reaction. We need that ATP to act as a phosphate donor because we need that phosphate on our full-meal glycinamidine ribonucleotide to act as a good leaving group, okay, because to allow for that kind of nucleophilic attack, that intramolecular nucleophilic attack that's going to govern cyclization of our full-meal glycinamidine ribonucleotide into our amino-imidazole ribonucleotide. Okay, so ATP itself, the phosphate group, is donated to that form amido oxygen. Okay, so that oxygen at the very end, that double bond oxygen that's linked to that amine group, that NH group, is going to have phosphate transferred onto it. An inorganic phosphate group is going to be transferred to it. Okay, and this essentially forms this kind of high energy intermediate, and this activates the amide group. Okay, and it kind of primes it for an intramolecular nucleophilic attack. So that amino nitrogen that's from the glycine that's kind of sticking out will attack that activated carbon on the form amido group, and this essentially results in cyclization. It closes that 5-membered ring and it forms that imidazole ring you see in purines. So we now have our 5-amino-imidazole ribonucleotide, and we got that for our full-meal glycinamidine ribonucleotide, okay, using ATP to activate that amide group and to essentially govern that cyclization, that intramolecular nucleophilic attack between that amide carbon and that amine group to allow for that cyclization and to form our imidazole ring. Now the next protein complex is a bifunctional protein complex. It only has 2 active domains, and I'll be talking about the steps more specifically when we kind of move on to the next slide. But I just want to make everyone aware that this is another multifunctional protein complex, but it's bifunctional, unlike our GART protein complex that was trifunctional and had 3 domains. Okay, and this protein complex is called phosphoribosyl amino-imidazole succinocarboxamide synthase, or PAIC for short. So the first step is a carboxylation reaction. Okay, so the enzyme that catalyzes this is the amino-imidazole ribonucleotide carboxylase enzyme, and that's part of our PAICS complex. Okay, so a carboxyl group is added directly to the imidazole ring of our amino-imidazole ribonucleotide. Okay, and that introduces the carbon that will ultimately become the C6 of the purine ring. Okay, that you see in purines. In particular, we say IMP because IMP is the first fully formed purine intermediate to our adenosine and guanine. Now, the carboxyl group comes from cellular CO2 or bicarbonate or HCO3, and that's fixated onto this aminoimidazole ribonucleotide to give you carboxy aminoimidazole ribonucleotide. And you can see that COOH, or that carboxylic acid functional group, now coming out of the imidazole ring. Okay, so now we go from carboxyaminoimidazole ribonucleotide to succinyl-aminoimidazole carboxamide ribonucleotide, or SAI, SciCare for short. Now, the enzyme that catalyzes this reaction is succinyl-aminoimidazole carboxamide ribonucleotide synthetase, and it's also called phosphoribosyl aminoimidazole carboxylase synthetase. So it has two different names in literature. Okay, and what we're doing here is we are adding an aspartate. The amino acid aspartate is acting as our kind of compound donor, if you like, and it's of course going to be an ATP dependent reaction, because you're going to need that you're going to need to stimulate a leaving group in order to fixate this compound onto our growing molecule. Now, the carboxyl group that was added in the previous step becomes activated by ATP, so a phosphate is donated to that carboxyl group, and this forms a intermediate. Now, that amine group on aspartate will attack that activated carboxyl group on the carboxy aminoimidazole ribonucleotide, and that aspartate acts as a nitrogen donor, and it becomes attached through that activated carboxylic group. Now we formed our succinyl-aminoimidazole carboxamide ribonucleotide. Okay, we need to remove that fumarate skeleton that we got from the attached aspartate. We're only interested in the amine group. The aspartate was an amine donor. Okay, so we need to remove that fumarate skeleton, and the enzyme that catalyzes this is adenylose succinate lyase, and this isn't a part of the PAICS bifunctional protein complex. This is its own enzyme that's working separately, and it's removing that fumarate skeleton, if you like. And as you know, with aspartate, when you remove that amine group, you get fumarate. So we only wanted aspartate as that amine donor. So once that fumarate skeleton's gone, we just have that amine group intact, and we get our aminoimidazole carboxamide ribonucleotide. So now we've removed that fumarate skeleton, or that aspartate skeleton, we've left our amine group, okay, on our 5-aminoimidazole 4-carboxamide ribonucleotide. The next steps is leading up to the formation of inosine monophosphate, which a lot of people will know is the first true metabolic precursor of our purines. It's starting to resemble that purine look. So we've got that imidazole ring, and you've got that 6-membered pyrimidine ring, and you've got that carbonyl group on the 6th carbon, which is very similar to what guanine has. As everyone will know, guanine and purine guanine has that carbonyl group on its 6th carbon. Adenine has that amine group, and once you get inosine monophosphate, the metabolism will either diverge and favor GMP synthesis, so guanosine monophosphate synthesis, or AMP, adenosine monophosphate synthesis. So, but before we talk about those kind of steps, we're going to talk about the next multifunctional enzyme complex, which will result in the formation of inosine monophosphate. So there's another multifunctional enzyme complex, and it only has 2 domains, so it's bifunctional, okay, and it's referred to as the 5-aminoimidazole 4-carboxamide ribonucleotide transformylase complex, or the inosine monophosphate cyclohydrase complex, and that, or ATIC for short. Now it has a aminocarboxamide ribonucleotide transformylase domain, which is involved in transferring a formyl group from our formyl donor N10 formyl tetrahydrofolate onto our aminoimidazole carboxamide ribonucleotide to form 5-aminoimidazole 4-carboxamide ribonucleotide. Okay, and then the next domain is an IMP cyclohydrase domain, and that's going to catalyze the intramolecular cyclization of the purine ring. It's going to cause that ring to close, and we're going to get our inosine monophosphate, which is the first precursor to our guanine and adenine nitrogenous bases. So we have our aminoimidazole carboxamide ribonucleotide. Now the next goal is to give it another carbon to kind of make that purine skeleton, because as you know, that heterocyclic pyrimidine ring consists of both nitrogen and carbon, just like the imidazole ring. So we get that carbon from our formial donors, our CHO donors, and that's our N10 tetrahydrofolate, or N10 formial tetrahydrofolate. The enzyme that catalyzes this reaction is the amino-imidazole carboxamide ribonucleotide transformylase domain. Okay, and that N10 tetrahydrofolate, as I said before, is used as a cofactor, and that formial group is transferred onto that exocyclic amino group. Okay, and this gives you your formial amino-imidazole carboxamide ribonucleotide, or FAICAR for short. Now, the next step is carried out by the IMP cyclohydrase domain. Okay, and that's going to catalyze the cyclization, or the formation of that pyrimidine ring that you see in your purines. So the nitrogen performs an intramolecular nucleophilic attack on that formial carbon. Okay, and this results in inosine monophosphate, and that contains the hypoxanthine base, if you like, and hypoxanthine is the nitrogenous base on IMP, and hypoxanthine is a precursor to both adenine and guanine. And now we're going to look at how they divert metabolically. So again, just chemically, as just another picture to kind of reiterate what's going on here, we start off with our amino-imidazole carboxamide ribonucleotide, okay, and we add that formial group from our N10 tetrahydrofolate, and that gives us our formial amino-imidazole carboxamide ribonucleotide, and now it's ready for cyclization. So IMP cyclohydrase is going to dehydrate this. Essentially, it's a dehydration reaction. Water's coming out, okay, and it's gonna govern that cyclization, and you're going to get the formation of IMP, and inosine monophosphate contains that nitrogenous base hypoxanthine, which is our precursor to adenine and guanine. So inosine monophosphate has two metabolic fates, if you like. It will either become AMP or GMP, so adenosine monophosphate and guanosine monophosphate. And of course, once you get adenosine monophosphate, you can undergo a stepwise phosphorylation to adenosine diphosphate. Then ribonucleotide reductase can then convert that sugar from a ribose to a deoxyribose sugar. Then when you have another stepwise phosphorylation process by your kinase enzyme, you get adenosine triphosphate, or deoxyadenosine triphosphate, I should say, and that deoxyadenosine triphosphate is a DNA-ready nucleotide monomer. It's ready for the polymerization. So you can kind of see the story kind of coming together now in this long pathway. So the enzyme that kind of catalyzes this branching, if you like, and this subsequent formation of adenosine monophosphate is adenylosuccinate synthetase. Okay, so what's happening now? If you look at the hypoxanthine, the nitrogenous base hypoxanthine, which is what's attached to that IMP, inosine monophosphate, the nitrogenous base that is incorporated into that structure is hypoxanthine. It's got a carbonyl group on its 6th carbon. Now, a lot of you will know that if you remember, adenine and guanine, they're both purines. They both have the imidazole ring and the pyrimidine ring. They're both heterocyclic, but they differ in their functional groups. So adenine has its amine group on its 6th carbon, whereas guanine has a carbonyl group on its 6th carbon, and guanine also has an amine group on its 2nd carbon. So it's really important to kind of remember those differences in functional groups, because it kind of helps you link these metabolic processes with their actual structure. Where are these functional groups actually coming from? So adenylosuccinate synthetase will use aspartate as a nitrogen donor to replace that carbonyl group, but in order to do that, it needs guanosine triphosphate, or GTP, as a phosphate donor to activate that carbonyl group, that 6th carbon, to as an electrophile, to be able to accept that amine group and to replace to that kind of electrophilic substitution reaction, if you like. So GTP is used instead of ATP for a very particular reason, because obviously we've used ATP a lot. Adenosine triphosphate has been used as a phosphate donor a lot in our metabolism. So to prevent adenosine adenosine triphosphate's been used as a phosphate donor a lot, and of course the byproduct of that is either adenosine monophosphate or adenosine diphosphate. And adenosine monophosphate buildup in cells is incredibly toxic. So to prevent adenosine monophosphate toxicity, this step uses GTP or guanosine triphosphate instead, because you'll get guanosine monophosphate as a byproduct. And similarly, when you look at IMP or inosine monophosphate branching into guanosine monophosphate, ATP or adenosine triphosphate is used as a phosphate donor to prevent guanosine monophosphate toxicity. So it's a very sophisticated metabolic process. It's very tightly regulated, and this is to prevent cell toxicity, because a lot of these intermediates, a lot of these byproducts, if they over accumulate, can result in toxic effects on the cell. But going back to this kind of branching to AMP from IMP, the adenylosuccinate synthetase will essentially transfer that phosphate group onto that carbonyl group on our inosine monophosphate, and this forms a intermediate 6-phosphoryl inosine monophosphate. Okay, it's very temporary, and that allows for that amine group in the aspartate to attack, and you get an electrophilic substitution reaction. Okay, and you get that aspartate on your hypoxanthine base. Now, the second step involves the removal of the fumarate skeleton, because as you know, we only want the amine group on that 6th carbon. We don't want the aspartate or the fumarate skeleton, and when you remove that amine group from aspartate, you get fumarate. So that adenylosuccinate lyase will catalyze step 2 as well, and it will remove that aspartate. It removed that fumarate skeleton, leaving that amine group on that 6th carbon, and now you have your adenine base. So as you know, adenine has that amine group on its 6th carbon, and that amine group was donated by the amino acid aspartate. And this reaction is essentially, I know there's a lot going on in this reaction, but it's essentially one reaction. These steps happen in milliseconds. But this is what's happening when you break it down mechanistically. You have to phosphorylate that carbonyl group to activate that 6th carbon as an electrophile, because this is in a substitution reaction where you're substituting that carbonyl group with an amine group. Okay, so you need to phosphorylate it to activate it, and then once you've fixated that aspartate onto your hypoxanthine base, that adenylosuccinate lyase will then remove that fumarate skeleton, leaving just the amine group intact, giving you your adenine nitrogenous base. So you now have adenosine monophosphate. Okay, and it can be phosphorylated again by like nucleoside kinase enzymes, or to give you adenosine diphosphate. And adenosine diphosphate is ready for your ribonucleotide reductase to turn that ribose sugar into a deoxyribose sugar, and then it will undergo another stepwise phosphorylation process to become a deoxyadenosine triphosphate, and it's ready for DNA polymerization. It's ready for DNA replication, okay, and daughter strand synthesis. So inosine monophosphate branch into guanosine monophosphate is slightly more complex than inosine monophosphate branch into adenosine monophosphate, purely because of that additional functional group. So we've already got, in our inosine monophosphate, we've got that carbonyl group on our 6th carbon, which is the same as what guanine has, but guanine has that additional amine group on its 2nd carbon. Okay, so that's and that might be a bit of a problem, because it's really hard to, there's no functional group there, there's just a hydrogen. As you know, it's really hard to replace say an amine group with a hydrogen. It's not feasible, it can't be done. So we first need to create a carbonyl group there to make that 2nd carbon more electrophilic. So the first step is to convert inosine monophosphate into xanthosine monophosphate, because xanthosine monophosphate has that carbonyl group on its 2nd carbon, and this is catalyzed by the enzyme inosine monophosphate dehydrogenase, and that uses the cofactor nicotinamide adenine dinucleotide plus, or oxidized, or NAD. Now that NAD plus will oxidize that C2, introducing a carbonyl group. So that C2 now has a carbonyl group, and it's become xanthosine monophosphate. Now that carbonyl group creates like it creates a functional group more suited or more primed for a nucleophilic attack, because we want to use glutamine, the amino acid glutamine, as a amine donor, or as a nitrogen donor, if you like. So that's going to be acting as a nucleophile, and it's going to target that carbonyl functional group. So in step 2, what happens is that that xanthocene monophosphate is converted into guanosine monophosphate. Okay, but it needs ATP and it needs glutamine to do this. So the ATP is used to activate that carbonyl group on that second carbon that we've just formed in the previous step with our IMP dehydrogenase and our cofactor nicotinamide adenine dinucleotide. Now that essentially, what that essentially does is it leaves it primed and susceptible for that amine group from that glutamine to come in there and to essentially replace that carbonyl group. But there's a few steps that happen before that. As you know, glutamine isn't just going to give up its amine group very easily. You need that amide hydrolysis, or that amine hydrolysis, to happen. So that GMP synthetase will have that will have a domain that allows it to isolate that amine group from the glutamine, giving you glutamate, and then that ammonia will be channeled through that hydrophobic ammonia channel in that kind of protein structure, and it'll be transferred directly to that second carbon. Okay, and you'll get a nucleophilic attack, and that carbonyl group is replaced or substituted with an amine group. And as you know, we now have our guanine nitrogenous base. We have that carbonyl group on our sixth carbon, which we haven't had to do anything to, because both inosine monophosphate and guanosine monophosphate both have that carbonyl group on their sixth carbon, and then we have our amine group on our second carbon, which we got through a nucleophilic substitution reaction. Now, once we get our guanosine monophosphate, as I said before, it will need to undergo stepwise phosphorylation before it becomes DNA ready. But it will need to be phosphorylated to guanosine diphosphate in order for our ribonucleotide reductase enzyme to essentially convert that ribose sugar into a deoxyribose sugar to give us our deoxyguanosine diphosphate. Then we undergo another stepwise phosphorylation process via our kinase enzymes to give us our deoxyguanosine triphosphate. And once again, our deoxyguanosine triphosphate is ready for that nucleic acid polymerization. It's ready. It's a DNA ready nucleotide. So as I said before, our adenosine monophosphate and our guanosine monophosphate will need to undergo stepwise phosphorylation before they can become deoxyadenosine monophosphate or deoxyguanosine monophosphate. Now, this is a readily reversible reaction, this stepwise phosphorylation, because of you know, for homeostasis purposes, we cannot get a build-up of any of these molecules because it will be toxic to the cells. So ATP is used as a phosphate donor, but it's also a readily reversible reaction, because if the buildup gets too much, then the enzymes can transfer those phosphate groups from your GDP back to your GMP and vice versa. So you have AMP plus ATP gives you ADP plus ADP, and that phosphate can be transferred as and when, depending on the buildup of your monophosphate or your diphosphate. So the reason we want to phosphorylate is for our ribonucleotide reductase enzyme to convert that ribose sugar into a deoxyribose sugar. Now, this is a common exam question. Ribonucleotide reductase works at the diphosphate level, not the triphosphate or the monophosphate level. The substrate it uses, it can only process these sugars. It can only convert these sugars on diphosphate molecules, not mono, not tri, just diphosphate molecules. So that stepwise phosphorylation process is really important in removing that oxygen, if you like. Now, just a little bit of background on this enzyme and the mechanisms behind the conversion between ribose and deoxyribose sugar. As you know, our ribose sugar has that additional hydroxyl group. A deoxyribose sugar just has that hydrogen. It doesn't have that additional oxygen. But you can't just remove a hydroxyl group from a ribose sugar, because it will disrupt the whole structure. The ring will collapse, and it won't be good. Okay, you need to do it strategically, and this ribonucleotide reductase enzyme uses radical chemistry along with the help of NADPH or reduced NADP, which we got earlier on in our pentose phosphate pathway. So when we oxidized our glucose 6-phosphate into phosphoglucono or phosphogluconolactone, okay, we got our NADPH, like I said earlier, to essentially create a radical that can then be further oxidized and reduced to remove that oxygen to give us that ribose sugar, and then we get DNA, essentially DNA, or sugar ready DNA, that deoxyribose sugar. So now we're going to look at the synthesis of pyrimidines, and in my opinion this is a little bit easier to kind of conceptualize because we're not using the phosphoribosyl pyrophosphate as a sugar scaffold. The rings are assembled, again they're assembled de novo, but they're assembled freely, and they're then attached to the phosphoribosyl pyrophosphate once they're fully assembled. Whereas with the purines, we saw the imidazole ring was built step by step, the imidazole ring, then the pyrimidine ring. Okay, and this is because the pyrimidines are much more simpler in structure. They're more simplistic in structure. Assembling them free floating is just more feasible. Now, but before we talk about that, I'm going to talk about this next kind of multifunctional or trifunctional protein complex, and this is called the carbamoyl phosphate synthetase complex. The first step in pyrimidine synthesis is the formation of carbamoyl or carbamoyl phosphate. Okay, I can never say that word. Apologies, guys. It doesn't roll off the tongue very easily. So the CAD enzyme has 3 domains. Okay, in eukaryotics, it has 3 domains. Okay, the first domain is the C domain, or the carbamoyl phosphate synthetase domain, or CPS2 type 2. The second domain is the aspartate transcarbamylase domain, or the ATCase domain, and the third domain is the dihydrorotase domain. Now, the dihydrorotase domain is really important in the cyclization. The carbamoyl or the transcarbamylase domain is really important in incorporating that aspartate in, and then the carbamoyl phosphate synthetase domain is that first domain. It is really important in that forming that carbamoyl or carbamoyl phosphate. I mean, this step tends to trip people up a lot because there's a few chemical equations that you have to remember to kind of understand what's going on mechanistically, but I've got them all down here for everybody's benefit. So the first step of pyrimidine synthesis is the formation of your carbamoyl phosphate. Now, as I said, this is catalyzed by the enzyme carbamoyl phosphate synthetase, which is the first domain of the trifunctional CAD protein complex. Now, first of all, ATP is used as a phosphate donor to activate CO2, and that CO2 comes from bicarbonate or HCO3, and this forms carboxyphosphate, and carboxyphosphate is what's called a high energy intermediate, so it's very reactive. Now, glutamine is acting as our nitrogen donor, if you like. So glutamine itself, as you know, we're only interested in that amine group, so we want to hydrolyze that amine group away from that structure. Okay, so this enzyme contains an amine hydrolysis subdomain, if you like, and that essentially isolates, forming ammonia, and it's tunneled again through those ammonia channels in the protein to prevent cellular toxicity, and then it's essentially fixated onto carboxyphosphate, and this forms carbamate, and this carbamate is another intermediate. Okay, and inorganic phosphate then leaves. Okay, so carbamate is then phosphorylated by a second ATP, forming carbamoyl phosphate, and ADP is a byproduct. So the initial phosphate that was used to activate the CO2 acts as a leaving group, allowing for that amine group from the glutamine to fixate, and then that leaving group goes, but then we have to phosphorylate it again to form our carbamoyl phosphate, because we need that. It's important we have those phosphates on, okay, to make sure our intermediates are metabolically active. So I'm going to break down the equations that I've got so we can see what's happening mechanistically, and if you remember these equations, it helps you kind of understand the process. So we've got our bicarbonate, or HCO3, and that's our source of CO2. Okay, and we've got our phosphate donor, which is our adenosine triphosphate. So that phosphate donation is going to give us carboxyphosphate, and we're going to have adenosine diphosphate as a byproduct. So it's only that gamma phosphate that's going on to the CO2. Okay, and then in a separate equation, we've got our glutamine, which is our amino acid, and then we've got our glutamine hydrolysis, if you like, via our carbamoyl phosphate synthetase 2 protein complex, and that's going to give you glutamate plus ammonia, so NH3. And that glutamate we're not really worried about, it's that ammonia that we're interested in. So that carboxyphosphate is that kind of high energy intermediate. It's going to be very reactive, and that phosphate's a good leaving group. So it's going to allow for that amine group to perform that kind of nucleophilic attack, if you like. Okay, and this will give us carbamate and inorganic phosphate. So we've got our amine group attached to our CO2 and our inorganic phosphate's gone. So then our ATP, again, another molecule of ATP is going to act as a phosphate donor, donating that gamma phosphate to form carbamoyl phosphate and ADP as a byproduct. So the overall equation to this reaction is glutamine plus CO2 plus 2 ATP gives you carbamoyl phosphate plus glutamate plus 2 ADP and 1 inorganic phosphate. And again, that inorganic phosphate has left from the formation or from the nucleophilic attack of the amine group from our glutamine to our carboxyphosphate, giving us carbamate, and that's where that inorganic phosphates come from. But mechanistically speaking, there's a lot of steps, and I don't know exactly who needs to know what, depending on what exam you're doing, what university is teaching you. But the main takeaway is the first stage of pyrimidine synthesis relies on the formation of carbamoyl phosphate, and that's catalyzed by carbamoyl phosphate synthetase, which uses ATP to activate our CO2 to kind of form our carboxyphosphate, and then that amine group from glutamate will form that that carbamoyl phosphate. Okay, so to kind of break it down, those steps that I kind of mentioned with the whole carbamate formation of inorganic phosphate, usually those mechanisms some courses don't necessarily need to know them, but it's good to know it so you can understand what's going on in terms of the steps. So the next step is we've got our carbamoyl phosphate, and we used our glutamine as a nitrogen donor, and we had CO2 and we activated that with phosphate, and then when that ammonia from the glutamine, when that NH3 group performed that kind of attack on our carbamate intermediate, our carboxymate intermediate, we got carbamate inorganic phosphate. We phosphorylated it again to get carbamoyl phosphate. So we don't need ATP in the next step because our carbamoyl phosphate already has that phosphate leaving group. Okay, so it's already primed for a chemical reaction. So the next step is the formation of carbamoyl aspartate. So we want the aspartate skeleton, if you like. Now, the enzyme that catalyzes this reaction is aspartate transcarbamylase, or ATKs. And in eukaryotes, as I said before, it's part of that CAD protein complex, that trifunctional protein complex. So the nitrogen on that aspartate attacks the carbonyl carbon on the carbamoyl phosphate. Okay, and a new C-N bond is formed, and the phosphate group leaves. So we have one nitrogen from glutamate and we have one nitrogen from aspartate, and both of those nitrogens will become a part of this pyrimidine ring that you see. Because remember, these rings are heterocyclic, they have nitrogen and carbon in their structure. So now we've got our carbamoyl aspartate. Okay, and we got that in the previous step when we had our carbamoyl phosphate, and that aspartate, that amine group, came and attacked that phosphate leaving group, and it allowed for that aspartate skeleton to be incorporated into our carbamoyl structure. Now the next step is a cyclization reaction again, closing of the ring. Remember, we're only forming a pyrimidine ring here, so the steps from chain to ring are much shorter because, as we said, pyrimidines are just that 6-membered ring. Now the enzyme that catalyzes this reaction is dihydroxyrotase. And again, in eukaryotes, it's part of that CAD protein complex, that trifunctional CAD protein complex. Now, the enzyme performs a dehydration reaction, okay, and what's happening is an intramolecular nucleophilic attack. The nitrogen from the carbamoyl group attacks that carbonyl carbon from the aspartate, and a new C-N bond is formed, and that 6-membered ring becomes closed, and you get H2O is eliminated. Well, even though we've got our six-membered ring, we haven't actually got an aromatic ring because it's not been fully oxidized, and we get the oxidation of dihydrotate into orotate in the next step, and that's a bit more of a nuanced step that I'm going to explain in the next slide. So this is quite a unique mechanism. This is quite a unique part of pyrimidine synthesis, and it kind of shows you how pyrimidine synthesis is actually directly linked to mitochondrial respiration, or cellular respiration, and indirectly contributes to ATP production. Now, dihydrotate is oxidized into orotate, and orotate is a structurally complete pyrimidine base precursor, if you like. Now, the enzyme that catalyzes this oxidation reaction is dihydrotate dehydrogenase, and it uses a flavin mononucleotide cofactor complex. And that flavin mononucleotide cofactor complex is essentially acting as an oxidizing agent. Okay, so it's capable of taking or accepting 2 electrons and 2 proteins from dihydrotate. Okay, so it itself, that flavin mononucleotide cofactor, will become reduced. Now, a lot of people might be familiar with the electron transport chain. So the electron transport chain plays a really crucial role in oxidative phosphorylation, which is essentially the final or ultimate phase of cellular respiration. It's where the most ATP is generated de novo without using or investing ATP into the process. So it's ATP generation without investing ATP, which is obviously a vital process to cells and cellular respiration. Now, it relies on a series of redox reduction reactions through these proteins embedded in the inner mitochondrial lipid bilayer, okay, such as complex 2, cytochrome C oxidase, ubiquinone, okay, and these protein complexes will take electrons. Typically, these electrons come from coenzyme complexes FAD and NAD, which are mainly generated in the Krebs cycle and glycolysis. Okay, but in this case, we've got electrons that have been donated from our dihydrotate, okay, and accepted by our flavin mononucleotide cofactor. So when our flavin mononucleotide cofactor, it will donate those 2 electrons to complex 2, okay, it itself will then become oxidized, and that complex 2 will become reduced, and then that complex 2 will donate those 2 electrons to cytochrome C, okay, and that cytochrome C will become reduced, and that complex 2 will become oxidized. Okay, so it's a series of redox or oxidation reduction reactions, okay, and that redox chemistry allows for the chemical potential energy to drive these hydrogen protons down an electrochemical gradient down the inner mitochondrial membrane, okay, giving ATP synthetase the energy, the chemical potential energy to synthesize ATP from adenosine diphosphate and inorganic phosphate. Okay, so that proton motive force, that movement of hydrogen down an electrochemical gradient, it's called chemoosmosis, essentially drives ATP synthesis. Okay, but what's driving that movement, that chemoosmosis of those hydrogen protons, is these redox reduction reactions. Okay, and dihydrotate is playing a role in that. It's indirectly contributing to ATP synthesis because that flavin mononucleotide complex, along with your dihydrotate dehydrogenase, has taken those 2 electrons, okay, and those 2 protons, and those 2 electrons will essentially be used by these like complex 2 and cytochrome C in redox chemistry to allow for that chemoosmosis to take place and de novo ATP synthesis to happen. And essentially those 2 protons will essentially indirectly contribute as well, because those protons are required to for chemoosmosis, those protons are required to move down that electrochemical gradient, okay, to essentially allow for ATP synthase to synthesize ATP. And of course, the electron transport chain is where the majority of ATP is synthesized, okay, and I won't talk about it too much because we're talking about de novo DNA, pyrimidine purine synthesis, so I won't get into it too much, but that just kind of shows you how dihydrotate is indirectly contributing to ATP synthesis, okay, and of course, in becoming oxidized, you get orotate, which is essentially your first fully assembled pyrimidine base precursor. Of course, orotate isn't used in your nucleic acids, but it is a fully assembled pyrimidine ring. So we now have our orotate, which we got through the oxidation of our dihydrotate, and that was directly or indirectly involved in the electron transport chain. So it now moves back into the cytosol. And before we kind of look at the next steps will result in the formation of our uridine monophosphate. Okay, and uridine monophosphate is the foundational. That uridine is the foundation for thymine and cytosine synthesis, if you like. So, but before we look at that, we're going to look at another kind of bifunctional cytosolic enzyme complex, another one of these protein complexes. And remember, it's important to visualize these enzymes as these complexes as multifunctional enzymes, because that concept this is what distinguishes us eukaryotic cells from prokaryotic or unicellular bacteria. Okay, or because they have a lot of these steps are controlled by different enzymes. They're enzymatically controlled in bacteria by single enzymes, whereas in eukaryotes we have these efficient protein complexes that allow for the kind of efficient synthesis of our purine and pyrimidine mononucleotide bases. Now, this bifunctional cytosolic enzyme is called uridine monophosphate synthetase, and eventually orotate becomes erythidine monophosphate. So it's actually fixed onto your phosphoribosyl pyrophosphate by your phosphoribosyl transferase or rotate phosphoribosyl transferase domain, and then you have your erythidine 5-monophosphate decarboxylase domain, which is where that carboxyl group at the C6 is removed via that kind of spontaneous decarboxylation. So the first step in biochemistry informally is called sugar fixing. So we're fixing that orotate ring onto our ribose sugar. And as you know, as I said before, our purines are synthesized using that phosphoribosyl pyrophosphate as a scaffold to kind of assemble that imidazole ring, then that pyrimidine ring kind of systematically. But pyrimidine synthesis is completely different. The pyrimidines, we saw they were synthesized freely using that CAD protein complex to form our carbamoyl phosphate, and then our carbamoyl aspartate, okay, and then our dihydroorotate, and then our orotate, okay. And then now we're kind of looking at fixing that orotate, that pyrimidine ring, onto our backbone, onto our ribose sugar, and this is catalyzed by the enzyme OPRT, which is part of that UMP, uridine monophosphate synthetase type 2 complex. Now the enzyme, the OPRT stands for orotate phosphoribosyl transferase, okay, and that's part of that UMP synthetase complex. And what that does is essentially provides it essentially fixates that orotate onto that C1 of our phosphoribosyl pyrophosphate, okay, and you get that activated pyrophosphate leaves as pyrophosphate, as PPI, and you get that orotate fixed on, okay, and that gives you orotidine 5-monophosphate. Now, this is an ATP independent process, of course, because we've already got those phosphate leaving groups. We don't need to add phosphates. Okay, so we've already primed that C1 carbon for this kind of electrophilic substitution. And I've got the equation down there as well, that orotate plus PRPP, that's our phosphoribosyl pyrophosphate, gives you OMP, our erythidine 5-monophosphate, and then plus our pyrophosphate, which is left. So the next stage is the formation of uridine 5-monophosphate. Once we've got our uridine 5-monophosphate, we can begin to kind of branch out into our thymidine, the formation of our thymidine and the formation of our cytidine bases, or our thymine and cytosine bases. So erythidine 5-monophosphate, this is a very simple step and a very important step actually. OMP undergoes decarboxylation, and the enzyme that catalyzes this reaction is erythidine 5-monophosphate decarboxylase. Okay, and that's in the uridine monophosphate synthetase protein complex. That's the second domain, and that catalyzes this reaction. And it's that carboxyl group that is removed, that COO group is removed, okay, and no ATP, no redox chemistry, just decarboxylation. And it's one of the most efficient enzymes in biology for that reason. It's able to just remove that carboxyl group very quickly. And obviously without that enzyme, that reaction will probably take years and years and years to actually happen. Now, in order to start synthesizing our the bases we're familiar with in DNA, because as we know, uracil does not exist in DNA. It's present in RNA. So uridine monophosphate is really important in uridine monophosphate is an RNA ready nucleotide, if you like. So we've got our UMP, and if we're looking at, say, protein synthesis and that kind of transcription phase where our RNA polymerase is making that strand of RNA, if you like, complementary to our DNA template strand, okay, UMP is perfect and ready to go and polymerize and form our RNA, our part of our RNA, because as we know, uracil is incorporated in RNA. But in order to get our thymine and our cytosine, we need to undergo stepwise phosphorylation, because this process happens at a triphosphate level to give us our uridine triphosphate, and then we're now ready to kind of branch out into forming our thymidine triphosphate and our cytidine triphosphate. So now we're going to look at the synthesis of cytidine triphosphate from uridine triphosphate. Now, as you know, uracil and thymine have a much more similar structure than uracil and cytosine do. So cytosine essentially has that amine group on its fourth carbon, whereas uracil has two carbonyl groups. So we need to get rid of that carbonyl group and replace it with an amine group. So the enzyme that catalyzes this reaction is called cytidine triphosphate synthetase or synthase, and it uses glutamine as a nitrogen donor. So glutamine is our source for our amine reaction. Okay, and of course we need ATP as a phosphate donor, because we need to activate that C4 carbonyl carbon, that C4 carbonyl group, to increase its electrophilic capabilities, to allow it to perform an electrophilic substitution reaction. So the phosphate is transferred from ATP, giving us our ADP. Okay, and that phosphate group will form a intermediate. Okay, so it will form our phosphoryl uridine triphosphate, if you like. Okay, that's a temporary intermediate. And what happens is then, as you guys will be familiar with by now, that amine group from the glutamine, okay, is obviously hydrolyzed by this CTP synthase, that it has a amide hydrolyzed domain, and it will hydrolyze that NH3, okay, it will isolate that amine group and it will tunnel it through the protein, and it will directly kind of fixate it onto this phosphoryl uridine triphosphate intermediate, and you'll have an electrophilic substitution reaction, and that carbonyl group will be replaced with our amine group, and essentially you get your cytidine triphosphate. Now, just to kind of go through those to kind of go through those equations, because again, in exams you may be asked to draw these equations out so you know what's happening mechanistically. So I've got UTP plus ATP, and that gives us our 4-phosphoryl UTP. So what have we done? We've phosphorylated that fourth carbon where that carbonyl group is. Okay, we've created a high energy intermediate. Okay, we primed our fourth carbon for an electrophilic substitution reaction, and we've got adenosine diphosphate as a byproduct. Then we're going to go down to glutamine. What's happening with glutamine? Well, our CTP synthase enzyme has an amide hydrolase domain, so it's going to, or an amine hydrolase domain, sorry. So it's going to isolate that amine group from the glutamine, giving you glutamate and your NH3 ammonia. Now, of course, that ammonia is not free ammonia. It's going to be traveling through a protein channel, a hydrophobic protein channel, to prevent cell toxicity, and it's going to be channeled through to the, I guess, the transfer domain, if you like, where that amine will act, and there will be an electrophilic substitution reaction where that amine group will come for that carbonyl group and it will replace it, and on that fourth carbon you now have your cytidine triphosphate. We've got that amine group now with cytosine. So the reaction itself is UTP plus glutamine plus ATP gives you cytidine triphosphate plus glutamate plus adenosine diphosphate, and of course that inorganic phosphate group that was transferred to our C4 carbonyl carbon in our uridine triphosphate is going to leave once that amine group replaces that carbonyl group in an electrophilic substitution reaction. Now, just to kind of reinforce the idea of nucleoside diphosphate equilibrium or kinase equilibrium, our cells maintain the balance between di and triphosphates. So what's going to be happening is ATP is going to be used, or ADP is going to be used to accept one of those, to transfer one of those phosphates from cytidine triphosphate onto ADP. So you get your cytidine diphosphate, depending on the level of cytidine triphosphate and cytidine diphosphate ratio. And of course that reversible reaction, okay, and readily available ATP allows for ribonucleotide reductase to kind of work at that diphosphate level, because obviously if you've got your cytidine triphosphate and then you've got your adenosine diphosphate, okay, nucleoside diphosphate kinase will transfer the gamma phosphate on the cytidine triphosphate to the beta phosphate on the adenosine diphosphate, forming that kind of phosphate anhydride bond, giving you adenosine triphosphate and cytidine diphosphate. And then when you get cytidine diphosphate, you can then, ribonucleotide reductase can then obviously work at the substrate level, and it can convert that ribose sugar into a deoxyribose sugar, okay, and you get your deoxycytidine diphosphate can undergo another stepwise phosphorylation process by kinase enzymes and becoming deoxycytidine triphosphate, and it's a DNA ready nucleotide, if you like. And you can see I've indicated that reversible reaction with two arrows going either way. You've got your CDP plus ATP, okay, nucleoside diphosphate kinase will transfer that gamma phosphate from the ATP onto our cytidine diphosphate when cytidine diphosphate levels, or nucleotide diphosphate levels, are increasing in the cell, okay, to give us more cytidine triphosphates and less cytidine diphosphates, okay. And it's a reversible reaction, because then once you kind of get that cytidine triphosphate, you can then get an ADP molecule, okay, adenosine diphosphate molecule, and that can become a phosphate acceptor, and that nucleoside diphosphate kinase can readily transfer that phosphate from your cytidine triphosphate to your adenosine diphosphate, okay, transferring that gamma phosphate onto the beta phosphate of the adenosine diphosphate, giving you adenosine triphosphate. And it essentially goes round in a cycle, and the cells are doing this they're doing this for many reasons. One, for equilibrium, to make sure you don't get toxicity, and two, because obviously we need our ribonucleotide reductase. You kind of take two birds with one stone. We need our ribonucleotide reductase to kind of work on that ribose sugar to make it a deoxyribose sugar ready for nucleic acid polymerization. Now, thymidine diphosphate synthesis is slightly different to the previous cytidine triphosphate synthesis, because thymidine diphosphate synthesis happens using the DUMP pathway, or the deoxyuridine monophosphate pathway. So uridine diphosphate must undergo the sugar must be changed to a deoxyribose sugar. So ribonucleotide reductase will come and work at the diphosphate level with uridine diphosphate to give you your deoxyuridine diphosphate, and then another phosphate group is removed to give you your deoxyuridine monophosphate. And thymidine can only be synthesized through the DUMP pathway, or the dump pathway, and this is because thymine is synthesized only in its deoxy state. And it makes sense, because a lot of you, if you know your RNA and DNA, you know that thymine is only present in DNA, therefore it doesn't really exist in a ribose state, because it's not going to be used. And as I said before, uracil and thymine are very similar in their molecular structure as nitrogenous bases. They both have those carbonyl groups on their carbons, but on their fourth and second carbon, but thymine has a methyl group on its fifth carbon, and uracil doesn't. So essentially what we want, once we get our deoxyuracil monophosphate, all we need to do is add a methyl group to that fifth carbon, and we get our deoxythymidine monophosphate. And obviously that deoxythymidine monophosphate will still have to undergo stepwise phosphorylation to be DNA ready nucleotide, but it doesn't have to undergo ribonucleotide reductase processing, if you like, because the ribonucleotide reductase would have done that when it was a uridine diphosphate. So essentially what's happening in this reaction is a methylation reaction. Okay, so only after the ribose sugar has been reduced, the enzyme thymidylate synthase will use folate or methylene tetrahydrofolate as a cofactor. That's acts as our methyl donor. In particular, it's called N5N10 methylene tetrahydrofolate. Now, the thymidylate synthase contains the amino acid cysteine in its active site, and that attacks that C6 carbon of uracil, and this activates or primes that fifth carbon for a nucleophilic attack. It forms this enzyme DUMP, or enzyme deoxyuridine monophosphate covalent adduct, between that 6th and 5th carbon, and that allows for that methyl group to kind of transfer from our tetrahydrofolate. So our tetrahydrofolate becomes oxidized to dihydrofolate, and that methylene group is taken off and placed onto that 5th carbon, and that gives you thymine, or that in particular deoxythymidine or deoxythymidine monophosphate. So you go from deoxyuridine monophosphate to deoxythymidine monophosphate, and then deoxythymidine monophosphate will undergo stepwise phosphorylation, another 2 stepwise phosphorylation processes by its respective kinase enzymes to give you a DNA ready nucleotide, thymidine triphosphate. So that's the end of my overview on the oxidative phase of the pentose phosphate pathway and the de novo synthesis of purines and pyrimidines. Now, I think of all the metabolic kind of processes to kind of learn when you're doing biochemistry, especially if you're doing medical biochemistry, or if you're doing biochemistry for pharmacologists, I think it's one of the more trickier pathways to learn, not necessarily because of the mechanisms, just because of the amount of long names to remember, and you're looking at it, when you're looking at it holistically, you're looking at the synthesis of both purines and pyrimidines, which obviously, as you now know, will take a different route metabolically, a different kind of strategy the cells use to synthesize those mononucleotides differently. I think in terms of the pentose phosphate pathway, it's a lot of people find it confusing at first, but the oxidative phase of the pathway is a lot more simplistic than the non-oxidative phase. And the non-oxidative phase of the pentose phosphate pathway, you don't really need to understand when understanding de novo synthesis and the synthesis of that ribose sugar. Of course, the non-oxidative phase becomes much more important when you're looking at energy metabolism, glycolysis, and all that. And of course, I'll cover that. But for now, if for whatever reason, if your exam's probably just going to be on like nucleotide metabolism, then that learning the oxidative phase is a lot easier. Now, my advice to a lot of people would be to not rely on abbreviations. I know people tend to abbreviate, but in exam conditions and when reading literature, sometimes they'll use the full names of these chemicals. So like when you look at GAR, for example, glycinamide ribonucleotide, it's good to learn the full name rather than the abbreviation, because sometimes it might not be referenced as GAR. And of course, I recommend everyone learn those long enzyme names, and they're called different things in different literature. So it's a lot to learn, but of course, like, it's worth it in the end. And I just want everyone to I hope this has been really helpful. And I just want everyone to do well in their exams and to kind of, approach biochemistry with confidence, because I think that's really important. But yeah, let me know what you guys are studying, and yeah, I hope everyone has a good rest of the week, and I hope everybody wish everyone the best of luck in their studies.